Welcome back, everyone. So we're continuing on Chapter 6, Reproduction at the Cellular Level from our OpenStax textbook. So in our last video, we started out just basically taking a look at our understanding of what the genome is. So in this video, we're going to dive a little bit deeper into how cells reproduce. So we're going to get into the cell cycle and talk about the whole entire process, which includes two phases: the phase of interphase as well as the phase of mitosis. So we'll go through that in this particular video. So by the end of this video, you should be able to describe the three states of interphase. You should also be able to discuss the behavior of chromosomes during mitosis and how the cytoplasmic content divides during cytokinesis. Okay, so it'll actually be different if it's an animal cell versus a plant cell. Define the basically what happens during G0 phase and explain how the three internal control checkpoints occur at the end of G1, at the end of the G2, as well as the M transition during metaphase. Okay, so so this again, we're going to take a look at the cell cycle, which it's a cycle, so it'll be like a sequence of events that takes place. We're going to see how it's carefully regulated with these checkpoints and so forth. We'll also take a look at these checkpoints or how interphase is divided into three phases, and then how mitosis is divided into 5 stages, and then how the cell actually physically divides into 2 through a process known as cytokinesis. All right, so let's talk about the cell cycle. So the cell cycle involves basically the cell needs to grow and it needs to divide. Okay, so it's growing bigger, and mainly during that time, we particularly focus in on the contents of the nucleus during this time. So we're going to focus in on how the DNA replicates, and so that when the cell actually physically divides into 2, each daughter cell has an exact copy of that DNA. Okay, now we learned from it back in chapter, what was it, like 3, about the contents of the cell, like the Golgi apparatus and the rough ER and the smooth ER and all that. So we're really not going to talk about how that part of cell growth takes place, but just realize that this does, all of that is taking place during this process of the cell cycle. But we're more focused in on the DNA part. Okay, so a lot of students kind of forget that the cell is growing, and during the cell growth time is when it's actually, making more mitochondria and Golgi apparatus and things like that. Okay, so when we talk about the cell cycle, first off, it's a cycle. Okay, so you can see it goes over and over again. So when a cell finishes dividing, it can go and grow again and divide again if it's a type of cell that will constantly replicate. So some cells we constantly do replicate over and over again, such as our skin cells that will constantly get worn out. Same with like blood cells, things like that. And our cell cycle, it's divided in basically 2 phases. The first phase is called interphase, and it takes up about 90% of the cell cycle. Okay, so this interphase is when the cell grows and the DNA is replicated during interphase. Okay, so you can see that this is about 90% of the time it's in interphase. About 10% of the cell cycle is in mitotic phase. Okay, so this is when the replicated DNA will start to separate, and the cytoplasmic contents start to separate, and then the cell physically divides into 2. Okay, so that's only about 10% of the cell cycle. So 90% of the time it's in interphase, 10% of the time it's in the mitotic phase. All right, so we're going to go through the different phases of the cell cycle, including interphase as well as the mitotic phase throughout this video. And to start us out, we're going to watch an Amoeba Sisters video on mitosis. So this video will give us a great introduction into the different stages of interphase and mitosis and give us some good visuals. So as we go through, pay attention to the vocabulary that they use and so forth. Okay, and we'll go into detail after that.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells. And in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and remind you to stay curious.
Alright, so we're going to walk our way through the different parts of the cell cycle. So even though that video focused mainly on the mitotic phase, we're going to go through all of the phases of interphase as well as the mitotic phases together. So it's always a good idea, again, to maybe make a chart. So if you were to make a chart, you might want to do something where you have something like this. I'm just going to kind of freelance it here. And then divide it into maybe 3 columns. So the first column will be the name of the phase, okay? And then the second will be a description, and then maybe the third part would be a picture, okay? Again, I like to do pictures, you know, similar to like what I had you do for the cell parts, just because if you can draw a picture of what's happening during the phase, it'll be more easier for you to remember what's going on, okay? And then you can kind of divide it, and then you have like interphase over here, and then you have the mitotic phase over here, and then interphase you have G1, S, G2, and so forth, okay? So you can do something like that if you'd like, to organize your notes. And of course, it's completely optional. For the interphase part, you, instead of just, there's nothing really to draw, but what you could do is draw what the chromosomes look like. So for the first part, what is the G1 phase? Okay, so we're in interphase. So interphase begins right after the cells have divided. Okay, so at this phase, you've just divided the cell into 2. So now you have 2 daughter cells. Okay, so these 2 daughter cells are just divided. Okay, so at this stage, if this was a human cell, it would have 46 chromosomes in each one of these cells, okay? And so it's basically just a new daughter cell, okay? And so each one of the chromosomes looks, you know, like one of those linear chromosomes like that, okay? So over here, I would write my description that it's basically undergoing some growth after mitosis, okay? So it's just formed, you know, it's just undergone cytokinesis, so it's, you know, a little smaller. It needs, you know, some Golgi apparatus, it needs, you know, some mitochondria, so it's growing. The DNA right now is just in linear chromosomes, okay? So it just looks like a linear chromosome at this point. So I'll just draw like a linear chromosome like that, okay? The next stage is called the S stage, okay? And the S stage stands for synthesis, DNA synthesis stage. So this is when the DNA actually undergoes replication, okay? And we'll go into the, actually the details of replication, how that occurs in, I don't remember what chapter, but in the next couple weeks, okay? But for now, you just need to know that what happens is basically we take our chromosome that looks like that, and we make an exact copy of it. So it looks like that. Okay? So now this chromosome has the exact same DNA as this chromosome. So we call these 2 chromosomes, and they're held together by what we call a centromere, okay? So a centromere holds the 2 identical chromosomes together, and these 2 duplicated identical chromosomes are what we call sister chromatids. So sister chromatids are identical copies held together by a centromere, all right? So in my picture, I might draw a picture of sister chromatid pair. So it just looks like, I don't know, or you can draw an X. Kind of looks like that, right? So during the S stage of, or S phase of interphase, DNA replication takes place. So we take our chromosomes and we make exact copies of them held together with the centromere. And during that time, so that by the end of mitosis, we have to have, you know, 1 copy of our chromosomes going into one cell, the other going into the other, and so that's why we have to replicate it first, okay? Another thing that it takes place during the S stage of interphase is that the centrosomes are duplicated. So if you recall way back when we talked about the structure of the cell, Centrosomes, they are basically these little structures that will produce mitotic spindle. So mitotic spindle, they are these little fibers that are part of cytoskeleton that actually the microtubule part of cytoskeleton. They the centrosomes, they will actually, since we're animal cells, our centrosomes consist of a pair of centrioles, and they produce the mitotic spindle. And mitotic spindle is something that will be used later on in mitosis, which will hold on to the chromatids. So we don't have to talk about it just yet, but just realize that during the S stage in our cells, the centrosomes are being duplicated so that we can have 2 sets of them, so that each set can go to opposite poles, producing the mitotic spindle. Okay, so that's the S stage of interphase. So by the end of the S stage of interphase, our single chromosome has now duplicated and held together by a centromere. And then finally we enter the G2, which is the second gap phase of interphase, and that's just where we undergo our final preparations for mitosis. So the cell grows a little bit more, gets ready, prepares itself for mitosis. All right. So that is the 3 stages of interphase. So G1 or gap 1, S DNA synthesis, and G2, gap 2. Okay, and again for G1 and G2, think of growth, because that's what's happening. They're growing during those phases. Okay, and then S phase, think DNA synthesis phase. They're replicating their DNA. Okay, so next we're going to go into the mitotic phase. So 10% of the time of the cell cycle, the cell is actually undergoing the mitotic phase. And that mitotic phase is separated into 2 parts. We have mitosis. So in mitosis, it consists of 5 stages. In the video, they talk about 4 stages. Your book separates prophase into prophase and prometaphase, so that's why we have 5 stages. So mitosis is basically this series of events that take place to separate the nucleus from, or basically separate the contents of the nucleus. Cytokinesis is this physical separation of the cytoplasm into 2 daughter cells. Okay, so think of mitosis as the 5 stages we're going to talk about, and then cyto means cell, kinesis means dividing. So it's the actual physical dividing of the cell. All right. So we'll go through mitosis in detail, as well as cytokinesis. And again, just realize that the video, it talked about 4 stages of mitosis. Your book talks about 5 stages. Cells, it's a cell. Humans, we are the ones that kind of label things the way, so depending on the book, some books call it 4 stages, some books call it 5 stages. So just realize that your OpenStax textbook calls it 5 stages, so we're just going to go with that. Okay, so here are our 5 stages. So we have prophase, prometaphase, metaphase, anaphase, and telophase. Okay, so the Amoeba Sisters video just had prophase, metaphase, anaphase, and telophase. So they combined prophase and prometaphase as one category. Okay, so let's go through each in detail. Again, if you have that chart out, always a good idea to draw what's happening in each of the phases. And it doesn't have to be as detailed, but mainly you just want to see, kind of draw what is happening with the chromosomes during that time, and what's happening with the centrosomes and so forth. Okay, so let's take a look at prophase. Now from the Amoeba Sisters video, remember they said pro means before. So I like to think of pro as prepare. So I think of it as the preparing to divide phase. So during this particular phase, we have some things that are taking place. So here is our cell. And if you take a look at the nucleus is still intact. Okay, so you can still see the nuclear envelope present, but it's starting to break down. Okay, so over here you can see it's completely intact, but over here it's starting to break down. So that's one thing that's happening in prophase. The nuclear envelope is breaking down. Another thing that you're starting to notice is that the centrosomes, remember for animals, they consist of these little centrioles. They start moving to opposite ends of the cell. Okay, so they're starting to move to opposite poles. Okay, so eventually they'll be at opposite poles. So they're starting to move to opposite poles. So that's another thing that's happening in prophase. As they're moving to opposite poles, the centrioles and the centrosomes, whatever, it's the same thing. They're starting to produce mitotic spindle. So you can start seeing this, blue stuff coming out. That's the mitotic spindle. So it's a type of microtubule. Okay, so that's starting to emerge from the centrosomes. And then finally, you can start to see the chromosomes become visible. And the reason why they're becoming visible is because they're condensing. Okay, so normally when you look at a cell that's not dividing under a microscope, the nucleus looks like this. So basically I cannot identify individual chromosomes, but as it starts to, and it just looks like that, as they start to condense, you can start to see little spaghetti streams. So they start to become visible. Okay, so those are basically the four things that are taking place during prophase, or the preparing to divide phase. Okay, so the chromosomes are condensing and becoming visible. The nuclear envelope is starting to disappear and break down. The centrosomes are moving to opposite poles, and the mitotic spindle is starting to emerge. Okay. Moving into the next phase is prometaphase. So that's kind of the in-between. Okay, so it's still preparing to divide. So in prometaphase, you can see the nuclear envelope has almost completely disappeared. So it's completely broken down here. The chromosomes are almost completely condensed. The mitotic spindle has completely emerged and is starting to attach to the centromeres of the sister chromatids. Okay, so let me get my little pen out. Okay, so I'm going to draw a picture, a very bad picture, but I'm going to draw a picture of a pair of sister chromatids. So these are identical chromosomes attached by a centromere. Okay, and this centromere, there is basically these little protein complexes which we call kinetochores. So kinetochore is basically the site where the mitotic spindle will attach to. So this is like mitotic spindle, it's going to attach to these little kinetochores, which happen to be on the centromeres. Okay, so mitotic spindle attached to the kinetochores. All right, and so you can see right at the center of these X's here, that's where the centromeres are, and these little protein complexes, that's the kinetochores where the mitotic spindle attaches to. Okay, sorry for the drawing. All right, so now at the end of prometaphase, we are all prepared to divide. Okay, we have our mitotic spindle attached to the sister chromatids. The nuclear envelope is completely disappeared. Centrosomes are in place. Everybody's ready to go. So now we enter middle metaphase. So I like to think of metaphase, middle metaphase. So think of the M for middle. So what happens during middle metaphase is that this mitotic spindle is going to push and pull these sister chromatids to the equator of the cell. Okay, so these sister chromatids are going to be lined up right along the equator of the cell, which is known as the metaphase plate. Okay, and that's it. Okay, so basically they're all lined up in the middle. And it's really easy to identify that stage under a microscope because you can just see them. They're all lined up right in the middle. Okay. Next, we move into anaphase. And I always think anaphase away. So think anaphase away because this is where the centromeres, they start to split. And remember the centromeres, they were holding the sister chromatids together. So if there's no centromeres, then the sister chromatids no longer are held together. But the kinetochores are still present and attached to the mitotic spindle, which is attached to the centrosomes on the opposite poles. And so during anaphase, if there's no centromeres holding the sister chromatids together, the mitotic spindle starts to shorten. And as it's shortening, the sister chromatids are pulled away from each other. So that's why I think anaphase away. So they're moving away from each other to opposite poles. All right, so they're moving away from each other. And you'll actually see that in the under a microscope. You'll start seeing, and you'll see this in the lab portion. You'll see the chromosomes, they start looking like they're clumping at either end of the poles. Right? And then telophase, I think divide into two. Okay, so telophase, think it's divided into two. But it's basically what happens in prophase, it's the opposite. Okay, so in prophase, remember the chromosomes condense. Well, the opposite. Chromosomes start to decondense. They start to unravel. In prophase, the nuclear envelope breaks down. Well, the nuclear envelope starts to reappear in telophase. Okay, and you end up during telophase, you end up with two nuclei present. In prophase, you start having spindle, mitotic spindle forming. In telophase, you have mitotic spindle breaking down. Okay. In prophase, you have like the centrosomes moving to opposite poles. Here you just have like the centrosomes, they start to kind of contract and like start to break down. Okay, the spindle fibers kind of continue to push everything apart from each other and break down. All right. So there are our 5 stages of mitosis. So we start with the preparing to divide phase, prophase, and prometaphase. Then they're lined up in the middle during middle metaphase. Then the centromeres dissolve or break into two. So then the sister chromatids are pulled away from each other during anaphase away. And then they prepare to divide into two during telophase. So at the end of telophase, you end up with basically 2 nuclei inside the cell. Okay. So finally, we end up with cytokinesis as the second part of our mitotic phase. So in cytokinesis, with cytokinesis, we have cyto, remember, means cell, kinesis means dividing. So this is when we actually have the physical dividing of the cell into 2 separate daughter cells. And this occurs differently depending on an animal cell versus a plant cell. So we'll talk about this in a separate slide. Okay. So let's go ahead and do a visual connection question to make sure we understand the events. So which of the following is the correct order of events in mitosis? So the first possible answer, the sister chromatids line up at the metaphase plate, the kinetochore becomes attached to the mitotic spindle, the nucleus reforms and the cell divides, and the sister chromatids separate. Is that the correct order? So the sister chromatids line up at the metaphase plate, the kinetochore becomes attached to the mitotic spindle, then the nucleus reforms and divides, and then the sister chromatids separate? No. Okay, so how about B? The kinetochore becomes attached to the mitotic spindle. Okay. The sister chromatids separate. Okay. Then the sister chromatids line up. Well, why would they separate then line up? So that one's not going to work. How about the kinetochore becomes attached to the metaphase plate? Why would, wait, the kinetochore becomes attached to the metaphase plate. The sister chromatids line up at the metaphase plate. The kinetochore breaks down and the sister chromatids separate. The nucleus reforms and the cell divides. That doesn't really make sense. Why would the kinetochore become attached to a metaphase plate? So the metaphase plate is just the equator of the cell. So, I'm guessing it's D, but let's just make sure. So the kinetochore becomes attached to the mitotic spindle. The sister chromatids line up at the metaphase plate. The kinetochore breaks apart and the sister chromatids separate. And the nucleus reforms and the cell divides. Well, that one looks most promising. Though I think this one is opposite. So I think D is the correct answer. Okay. Okay, so even though it sounds kind of weird where the kinetochore breaks apart and the sister chromatids separate, I think they separate. I think they meant like the centromere breaks apart and then they get pulled to opposite poles. Okay, so D would be the correct answer there. All right, so moving on. So cytokinesis, again, is that second part or the last part of mitosis. So it is different in animal cells versus plant cells because plant cells have a cell wall, animal cells don't. So it has to be slightly different. So if we look at animal cells, when animals undergo cytokinesis, it starts with a pinching inward of the cell. And when it pinches inward, it forms what's called a cleavage furrow. Okay, so it just kind of pinches inward on either side of this elongated cell until the pinching goes all the way inward until you end up with two separate cells. Okay, so that's how cytokinesis works in an animal cell. In a plant cell, it's a little different. Instead of it being pinched inward, okay, you have this, big cell wall on the outside and then the cell membrane. It starts inwardly. Okay, so we actually have these vesicles that start to fuse together along the center of the cell. And then as they fuse together, they start moving outward until they seal it off completely. And that is what we call a cell plate. Okay, so it's these vesicles, the Golgi vesicles, they seal off into two separate cells. Okay, so it the cell plate seals it off until you end up with two separate plant cells. So cleavage furrow pinches inward, cell plate moves outward. Okay, so it's the opposite basically. All right, so that's how we end up with two separate cells in our animal cells versus our plant cells. Okay, all right. Now, moving on, we talked about the cell cycle in complete detail, starting with the G1, the S, and the G2, and then finally with the mitotic phase. Now, when the cell is not actively dividing, it will leave the cell cycle and enter what's called the G0 phase. So basically once it's done with mitosis, you have the two, daughter cells, it's going to enter possibly the G0 phase. So that's occurs when the cell is not actively preparing to divide. Now some cells will be there temporarily. So they'll, stay in this G0 phase until some sort of external signal triggers them to re-enter the cell cycle in G1. Okay. Some cells stay in the G0 forever and rarely leave it. Okay. So it just kind of depends on what type of cell. And we'll talk about, the variability. Okay. So just realize that once mitosis is over and the mitotic phase is over and you have two daughter cells, those daughter cells will leave the cell cycle and go into G0. And they could stay there forever, or they could re-enter the cell cycle and prepare to divide once again. Okay. So let's talk about the control of the cell cycle. So the length of the cell cycle is highly variable. Okay, so early on in embryonic development, so like when the cell sperm meets egg and you have a zygote, and then it starts to divide into 2 cells, into 4 cells, into 8 cells, and 16 cells, and you get clump of cells, a blastocyst, and you get a gastrula and blastula and gastrula and embryo. The average length it takes in the cell cycle is anywhere from 2 to 5 days of actively dividing. So, that can be, fairly average for early embryonic development. And that's for every single cell. So think about, if you have 4 cells that are actively dividing at the same time, all 4 cells are actively dividing, and you get, you're going to start getting clumps of cells pretty quickly. Some cells in your body, they may never divide again. Okay, so once you have, for example, some cardiac muscle cells, they will never divide again. So, and some neurons, they never divide again. So if you think about it, think about like a traumatic brain injury. If you lose function of that part of the brain, you're never going to gain it back, right? Because those cells are not going to divide again. So you can't replace them if you do damage to them. Same with spinal cord injury or something like that. You can't make more neurons. So that's why some of our cells we can repair easily. And if you get a cut, your body can heal pretty quickly. But other parts of your body that if it gets injured, it could be something that you're going to live with that injury the rest of your life. So think of like a severed spinal cord, something like that. Or traumatic brain injury, or if you take some really bad drugs and you really screw up your brain, it's going to be permanent brain damage. Okay, so take care of your brain is what I'm getting at, because some of your cells will never divide again. So you got to take care of those neurons and those cardiac muscle cells. Okay, so that's why we have to, some of those cells, they do actively divide all the time. Some of them, they never divide. Okay, so what about the cells that do divide? How do they know when to divide? Okay, and how do we prevent cells from dividing when they should not divide? Well, we have regulation of internal checkpoints throughout the cell cycle. So this will prevent a cell from dividing when it should not divide. Okay, So we actually have three of them. We have a G1 checkpoint, so that occurs during the G1 phase of the interphase. We have a G2 checkpoint that occurs during the end of the G2 phase. And then we have an M checkpoint, so that occurs during the middle metaphase of the mitotic phase. Okay, so the first one, the G1 checkpoint, that is also known as the restriction point. So this will stop the cell and check to make sure that the cell is supposed to divide. Okay, so it's making sure that the cell is actually good to go. Okay, that it's scheduled to divide, that it's supposed to divide, so that it should continue with the cell cycle. Okay, so if it receives the signal to go, it will continue onto the S stage. If it doesn't receive the signal, then it'll leave the cell cycle and go into G0. So it's the restriction point. It'll prevent cells that should not be dividing from going any further. Okay. So if it gets the signal to go, it'll continue going into the S phase where we have DNA replication take place. And then it'll continue to the G2 phase where we have another checkpoint. So this G2 checkpoint will check and make sure that DNA replication took place correctly, that all the chromosomes were replicated correctly, and that none of the DNA was damaged along the way. Okay, so that's kind of a DNA checkpoint. We got to make sure that all the DNA is correct, that the chromosomes look like they were replicated correctly, and that the sister chromatids are attached to their centromeres, and that everything looks good to go. Okay, so if that passes the checkpoint, then it'll continue on to the mitotic phase. So in the mitotic phase, we go through prophase, we go through prometaphase, we go into metaphase. Then we have the M checkpoint. So in middle metaphase, there's this M checkpoint called the spindle checkpoint. So this is where we stop and make sure that all the sister chromatids are correctly attached to the spindle microtubules. Okay, we don't want any chromosomes just like floating out there, not attached to anything. So we have to make sure that all the chromosomes are attached to their mitotic spindle at the middle of the metaphase plate in the middle of the cell, so that when anaphase occurs, they split and move to opposite poles. Okay. So we do have these three checkpoints just to make sure that everything's running smoothly. All right. Now, of course, sometimes things slip through these checkpoints, and then, we'll talk about that in the next video. But those, kind of assure that any errors or any, complications or anything that goes wrong can get fixed before it continues on. All right. Okay, so let's do some review questions. Chromosomes are duplicated during what portion of the cell cycle? Is it the G1 phase, the S phase, prophase, or prometaphase? So what portion of the cell cycle are chromosomes duplicated? And here's the sister chromatids, by the way. This is what it looks like. That would be during the S stage. So think synthesis of DNA phase. So that's the S stage or phase of interphase. Here's another question. Separation of the sister chromatids is a characteristic of which stage of mitosis? Is it prometaphase, metaphase, anaphase, or telophase? When do the sister chromatids separate? That's during anaphase away, right? So C, anaphase away. And then we have the individual chromosomes become visible with a light microscope during which stage of mitosis? Is it prophase, prometaphase, metaphase, or anaphase? So when do individual chromosomes first become visible with a light microscope? If you said a prophase, I would agree with you. So they continue to condense during prometaphase, but they first start condensing and become visible during a prophase. And then finally, what is necessary for a cell to pass the G2 checkpoint? Is it A, the cell has reached a sufficient size, B, an adequate stockpile of nucleotides, C, accurate and complete DNA replication, or D, proper attachment of mitotic spindle fibers to the kinetochores? So what has to happen for a cell to pass the G2 checkpoint? If you said C, accurate and complete DNA replication, I would agree with you. Okay, so this is where the cell has to, have completed DNA replication and that everything looks complete and accurate. Okay, D, that's the M checkpoint. All right, we are done with 6.2. Okay, the cell cycle. So by now you should be able to describe those 3 states of interphase, G1, S, and G2. You should be able to discuss the behavior of chromosomes during mitosis. So think about what happens during prophase, prometaphase, metaphase, anaphase, and telophase, and how the cytoplasmic contents divides during cytokinesis in both animal cells and plant cells. You should be able to define what happens during the G0 phase and how long it can last for some cells. You should also be able to explain how the 3 internal control checkpoints occur at the end of G1, the G2, and the during metaphase. All right. So in the next video, we're going to get into cancer in the cell cycle. So this is when we have some errors that occur, and that could lead to cancer. Okay.